Okay, ladies and gents, Toll Like Receptors. Quite an overwhelming topic, so I'm going to try and keep this as easy and simple as possible, and how we can remember which Toll Like receptors are on the cell surface and which of those are on the nuclear envelope in this particular situation. Now, before we go into that, what exactly are Toll Like receptors? Well, what we need to appreciate is that they are essentially PRRs. And what is a PRR? A pathogen receptor, recognition receptor. So this guy really essentially recognizes pathogen patterns. And what will they be? They'll be pathogen associated molecular patterns, or otherwise danger associated molecular patterns. So a PAMP essentially is the likes of an antigen. Now, if we go back a little while, you will have recalled an antigen and learnt that an antigen is a molecule, usually a protein that the body recognizes as non-self, or it recognizes it as foreign, and that therefore initiates an immune response. So that's what a PAMP is, okay? A pathogen associated molecular pattern. Now, what's a danger associated molecular pattern? So the difference between PAMP and DAMP, a danger associated molecular pattern is more endogenous. It's actually within the body. It's something that the body might create itself. So, for example, as a result of necrosis, as a prime example, some of the mediators that will be released are essentially recognized as danger associated. So DAMPs are endogenous and PAMPs are exogenous. Okay, so Toll Like receptors essentially will recognize these PAMPs and DAMPs, and therefore Toll Like receptors are your pathogen recognition receptors, really. So we have these TLRs on the actual cell surface, the outer cell membrane, and we have them inside on membrane-bound organelles, and in this given situation we have been taught those in the nucleus. So these are nuclear endosomal TLRs. We've been given 1 to 9, so we need to know all those that are, and the question is, well, which one is going to recognize what? And so what we appreciate is I'm going to go, and first of foremost I'm going to say, well, the nuclear ones, 3 is on the inside, and from 1 to 9, so 3 plus the last 3. Okay, so 3 plus 1, 7, 8 and 9 as the last 3. Okay, these are all on the inside, and because it's nuclear, we know the nucleus is associated with genetic material, and so we've got literally all our genetic material here. So next it's a matter of, okay, so what kind of genetic material? Well, the way I remember this is 3D. Things are 3D, so that's that one, and that's because it's double-stranded RNA, hence 3D. 7-8 single-stranded RNA. We've already dealt with the double-stranded, so here we'll do single-stranded, and your S for 7 starts off with 7-8, so single-stranded. And last but not last, we've got 9, and that's associated with G, and 9G are kind of similar in how you might write them, so that's how I remember CpG RNA for 9. Now moving on to the outer membrane. So here we have to appreciate that we have got a couple of dimers. Okay, so these are dimers, and di means 2 because there's 2 of those combined together, and these are heterodimers, and the reason they're hetero, the reason we're calling them hetero is because they are 2 different TLRs coming together. So here what we want to, there's no easy way to remember 1, 2 and 2 and 6, but what is useful here perhaps, and I'll use a different colored pen now, what is perhaps useful is the fact that 1 add 3 makes you try, so 1 add 2, sorry, so sorry, I was distracted by the pen, so 2 add 1 makes you 3, hence you try, and we've already got tri-acyl, so now we've got di-acyl as being the second of the 2, So there's two of them, two dimers, and each contains the number 2, and you've got a diacyl, so 2, 2, and 2. Next in line, and these are peptides, okay? This is important because the next in line is your 4, and 4 for L, and we write the capital L and make it into a 4. So 4 lipo, and this is a polysaccharide. And that so that's lipopeptide and polysaccharide, 4 followed by 5 because S looks like a 5. So last but not least, we've got number 5 and F for 5 and F. Notice how I wrote 5 like that and then F kind of like that, and that's associated with your flagellin. So there you have it. I hope that was helpful. We got an indication as to what TLRs actually are, what they recognize. They are also TLRs are your PRRs. They recognize pumps and dumps. Pumps are exogenous, whereas your dumps are endogenous. So exogenous in that they come from outside, and endogenous in that they are from within the body. Then you have TLRs on the outer cell surface, and the ones we've been taught are those on the nuclear envelope or the membrane, and those that are with these and those that are on the outside. Thank you for watching.